Thank you very much for Sergey for introduction of brief introduction about me. So I am not a polymer chemist. I am a pharmacist. I'm graduate of Kazan State Medical University, but my PhD work is dealing with polymers, with interpolymer complexes. And I'm very proud to say that my first publication is in Russian, of course, at that time in 19th of 20th century, is published together with great scientists in polymers. It's academician Kabanov, it's Professor Zezin, and my scientific supervisor is Professor Vera Alexandrovna Kemenova. And we published our first review. It's the name of application of interpolymer complexes in pharmacy. So, that way, at that time, previously, I was interested in polymer complexes, and during all my life, of my scientific life, of course, I'm dealing with this topic. So, of course, interpolymer complexes is well known. It's formed between two oppositely charged macromolecules. And we did, of course, but anyway, I should mention that Professor Nagai, which is yesterday mentioned by Professor Vitaly Khutaransky, and he said, of course, that he is one of the pioneers of bioadhesion studies. I should add that Professor Nagai group is also one of the first to publish the papers which involving of interpolymer complexes, and it not only interpolyelectrolyte complexes stabilized by ionic bonds, but it's also interpolymer complexes stabilized by hydrogen bonding. So both types of these polymer complexes is published by the group of Professor Nagai. So today I speak mostly about oidragids. So this is the pharmapolymers, and we did interpolymer complexes between them, between chemical complementary grades of it, mostly stabilized by ionic bonding. So why is pharmaceutical polymers? Of course, if we used new polymers, new synthesized polymers, so it was a problem that they could be or not, of course, have unpredictable toxicity. So when we use the pharmaceutical polymers, of course, we have not have such kind of problem. Evonik company, Evonik pharmapolymers, they did a lot of types of eudragid family polymers, and we use, of course, only some of them which has opposite charge groups. All the polymers are already used as a coating materials and could have a delivery to different parts of gastrointestinal tract. So some of them have polyion or polycation structure, and so of course we use it. We use them in our work. So a lot of classification of profiles which could be achieved by combination of the drugs with polymers, not only polymers but all the pharmaceutical ingredients. So and we have achieved different types of them, and it depends, of course, not only of composition of these types, but types of dosage forms, but also it depends from the physical chemicals properties of the drug and, of course, the used polymers. So we have to see that there are a lot of problems of using a drug polymer in matrix drug delivery, not in coatings, but in matrix drug delivery. So due to copolymer structure, some of gastro-resistant polyanions, when it was formed as a dosage form as a matrix tablets, they have disintegrating in the stomach acidic region. Gastrosoluble polycations is dissolving the stomach region. That is of course obvious. But when we use polymer mixtures, so physical mixture of polyanion and polycations, they also have ability to disintegrate, and that we think that maybe preparing of interpolymer complexes could have solved such kind of problem. One of the pioneers who think about this problem is Professor James McGinty from the University College of Pharmacy in University of Texas in Austin. He has think that when he tried to combine two oppositely charged eudragit copolymers, he said he mentioned that the profile of combining polymers, it's only a physical mixture, could slow up, and he thinks that maybe it could be the reason of it interpolymer interaction. But unfortunately, he has not proved it. He is only think about it as a hypothesis. So we have prepared interpolymer complexes between oppositely charged polymers, and originally existing matrix system that already exists in the pharmaceutical market is the Eudracol system in pellet dosage forms, and matrix tablet, which is named CODES. We are developed some of them, so it's not only oppositely charged eudragit, but also combination of eudragit with sodium alginate, pectin, or combination of polycation eudragit EPO with counter-charged carbopoles, or combining and preparing the polymer complexes between eudragit and high Kazan. But in frame of this presentation, I'm mostly focused only of interpolyelectrolyte complexes, which is formed between two oppositely charged eudragits. So the first one is the group of interpolymer complexes that we developed for colon targeting drug delivery system. And one of the pioneer of it is Professor Abdul Basit, who has a presentation with interesting title. It's a dirty job, but somebody has to do it. So we also did a lot in this frame of this topic. So we developed tablets and also microencapsulate forms. And we also examining how to deep insight experiments in order to know what has happened during swelling and drug release process. So we have did swelling experiments and tested the swelling in simulating gastric fluids and simulating intestinal fluid, and separate the gel layer and also investigate the core of it. And we did it not only in matrix, but also in microparticulate drug delivery systems, and also to check the interaction between two layers, which is formed in microencapsulation process. So the first, as I already told, original drug delivery system, which is called Eudracol, which is done by Vonik. So they had very simple structures. So it's non-parallel sugar sheets. There are a matrix layer and two coatings. The inner coating is a combination of polycation eudragit types, RL and RS, and outer layer is colon targeting eudragit type, is eudragit FS30D. So, you see the profile of it compared to the traditional one, and pharmacokinetic profile also, pharmacokinetic curve, which is proved that it was sustained release, and not only sustained, but it's delaying to the colon region. And we see very interesting study which is published together with American and German scientists of the University of Georgia and Evonik scientists, and they uh, try to investigate what has happened if this Eudrakol system is prepared. And they published very interesting paper about the investigation of potential ionic interaction between anionic and cationic polymethylcrylate types of novel colloid drug delivery system, but they have think that, of course, it's oppositely charged and could be interact, but they not find the interaction between them due to they tested it only in modulating process of ink manufacturing. And we think that the interaction could be happened, of course, but it's happened during release process. So, and we did the same work and we prepared the same bilayer structure of Eudrakol system and examined it by two methods, by Fourier transform infrared and DSC, and found that there are interactions between two polymers within and during the process of releasing it. The second system is CODAS, is also have two coatings, and one of them is Eudragit E100 and L100, so they are oppositely charged, as you see, and they could also be interact. And we are also interested about this system and published a paper, that our first paper in international journals, together with collaboration of Leuven Katalin University with Professor Guy van der Mutter. And we checked that interaction between these polymers happened during the release process, and we prepared such interpolyelectrolyte complexes in order to see how it works and use the model drug of ibuprofen. So we also did we choose another polyanion, it's L155, and we also prepared the systems by interaction polycation and polyanion of Eudragit family copolymers, and use as a model drug is diclofenac sodium. So together with Professor Sermina, she is from the Department of Pharmacology of our university, we did comparative in vivo and vitro assessment of these polycomplexes drug delivery systems on rabbits. And we see that it was good correlation between the release profiles and pharmacokinetics curves, and combining different composition of interpolymer complex that we prepared, we have a different pharmacokinetic curves, which could be delayed release or slow release, sustained release pharmacokinetic curves. We also did, so we also choose another one, polyanion, it's S100, and also prepare the polycomplexes with them, and also test it on the rabbits and see that it was also colon targeting system. Previously, the Evonik specialists, scientists, announced that they have also interested in interpolymer complexes preparing, and they prepared it in organic solutions. So first it announced at AAPS World Meeting in 2007. So, and published a nice paper about such kind of combination of oppositely charged Eudragit polymers. And we also did such kind of work. Unfortunately, our colleagues in Germany have a conclusion that such kind of complexes are not useful as drug delivery carriers due to its disintegration, disintegrating, simulating gastrointestinal fluid. But we find how to solve this problem, and our complexes are not disintegratable, so they are good swelling, good eroding during the release process, but not disintegrating. So, and we published papers, so it finds that the interaction between this type of copolymers in organic solutions, we use mixture of organic solutions, ethanol, tetrahydrofuran, and finds that such kind of interpolymer complexes are stabilized both by ionic bonds and also hydrogen bonding. So, and this is, of course, very good conclusion that because preparing such kind of complexes have made the application more technologically relevant. We also test such kind of system in biorelevant media in order to see, and as you see, it's very compatible to buffer solutions and biorelevant media. There are very close results. Then we also, together with University of Salerno in Italy, we did very interesting work of microencapsulation and choose countercharge eudragids and also combining with countercharge L-ginatsurium and prepare microencapsulation and used indomethacin as a model drug. And also we did another work and choose model drug of 5-fluorouracil and prepared such kind of system too. We also prepared indomethacin containing interpolyelectrolyte complexes based of oppositely charged eudragids and involving indomethacin as a drug, which is also could be interactable with polycation. In this case, it's eudragid EPO in prepared 3-component interpolyelectrolyte complex, which include not only 2 countercharge polymers, but also a drug. We also examined that hot bed extrusion. This is a technique which is used also in pharmaceutical industry. We prepared hot bed extrusion interpolymer complexes with low TG, so we could. It's also be applicable for that purpose, and we did this work, but not already published. It's only in the preparation of publication. So we did a nano-sized macroparticulate system and matrix drug delivery system by using our equipment. So this equipment is we have in our lab, and this is some kind of scheme how to prepare it in our lab. So we also did gastro-retentive systems, which is floating on stomach fluid, and it works like this. We also did some work and published already about the using of combination of oppositely charged eudragid as a floating material in order to prepare floating drug delivery systems. We also did some work about the buccal drug delivery system and or dispersible systems, which is also where I did with University of Milan and with University of Reading, and published some works concerning this topic. If we think about the future perspectives about the using interpolymer complexes based on oppositely charged eudragid, so we also did a lot of, I hope so, in nearest future. So we are prepared interpolymer complexes by using another polycation eudragid RL. So together with Professor Vitaly Khutaransky, we are at the beginning of work concerning the chemical modification of eudragid structure and preparing pegylation system and fluorescent labeling polycation. About pegylation polyanion, Vitaly was already announced yesterday in his presentation. This is our new topic, so it's not concerned gastrointestinal drug delivery, it's transmucosal. And we have our mutual PhD student, Natalia Porfirieva, and we're happy that this year we receive foundation from Russian Science Foundation, so it's a project. And as a leader scientist in this project is involving Professor Vitaly Khutaransky from the University of Reading, and we also very happy that Professor Sergei Filippov is also joined us to this project, and I hope that we could do a lot of in order to prove that such kind of interpolymer complexation is also capable for delivery, intranasal delivery, nasal delivery to the brain, and using psychoactive drugs for this purpose, of course. We move to conclusions. So combining grades of oppositely charged eudragite gave a lot of possibilities to simply regulating the solution profiles. So the study demonstrates the potential of IPEX of using eudragite copolymer family polymers in order to have good careers for oral drug delivery systems, and could deliver the drugs to mouth, stomach, and different parts of the intestine, including colon. And also chemically modified eudragite copolymers is potential to be used as nasal transmucosal delivery to the brain. I hope so. And I'm very happy to announce our collaborations. It's Professor Anna-Angela Barber from University of Salerno. It's Professor Nicoletta Fataki from University of Bas. Of course, Professor Vitaly Khutaryansky from the University of Reading. Professor Giva Andermutter from Leuven. Francesca Celurzo from Milan, and Kairakis Kashrimanis from Greece, from Saloniki University. And of course, as already told, we are happy that Professor Filipov is also joined us and became our collaborator. And of course, I am happy to have financial support from Russian Science Foundation and Russian Foundation for Basic Research. And special thanks to Professor Zayin. Of course, unfortunately, he is gone already. Professor Bronich from University of Nebraska, and my scientific supervisor, Professor Vera Alexandrovna Kemina, and the members of my group, of course, which you see on the slide. I'm happy that they help me to realize not only my ideas, but it's of course ideas of young scientists, and that is very nice, and I'm very happy to have them. Thank you for your attention.